Hi everyone, I'm back with part 2 of Retina. So I've already covered part 1. Let's quickly summarize that first. Now, retina has epithelial cells, then you have the photoreceptor cells. By photoreceptor, we mean the cells that detect the light. That includes rods and cones. Then you have the neuronal cells. In neuronal cells come both the bipolar cell and the ganglions. And then we have the glial cells. So in the previous video, we discussed these three cells. In this video, let's cover the glial cells. Now, glial cells are the cells that provide protection, support, and nutrition to neuronal cells. Now, your nervous system, it has two main types of cells, neuronal and glial cells. We hear about neurons every time, but we have to give equal credit to glial cells also because they provide support and nutrition to your neuronal cells. Right, so we'll discuss them in detail. Before that, let's go over a quick summary. So this is showing the three layers of eyeball. So you have the outermost, which is the protective layer, that's sclera, and then you have choroid, which comes in the middle layer, and this is the nutritive vascular layer. Then you have retina. Now, retina is the innermost sensory layer and it is made up of two parts, the pigmented layer and neural layer. Now, pigmented layer is the 10th layer of retina, and as I've already told you, that retina is made up of 10 layers. So, pigmented layer is the 10th layer, and neural layer comprises of the remaining 1 to 9 layers of the retina. Now, this is the microstructure of retina. First of all, in black is the sclera. Then you have choroid. So posterior ciliary artery comes and then it forms vascular loops. These vascular loops have perforation to allow, the food substances like nutritive substances to basically seep out. And they are called choriocapillaries, or even you can call them sinusoids. Sinusoids because they're slightly bigger than capillaries. Then you have the pigmented epithelium. Now, pigmented epithelium here you can see that it has tight junctions in the middle, and there's great importance of this that we'll cover later. Other than that, these obviously have nuclei, and very important structure here. They have pigments called melanosomes. These melanosomes they basically absorb the extra light, and this prevents any sort of internal reflection. Therefore, whatever image that you're going to form will be perfect, and it won't be degraded in any way, right? So these are the epithelial cells. After that, we have the photoreceptor cells. That in photoreceptor cells we have rods and cones. Now, uh, rods and cones. Let's discuss their structure in detail firstly. So this is a rod, and rod has first of all an outer segment. Outer segment is the photoreceptive part. Then it has the inner segment, and then you have its nucleus, and then you have the synaptic vesicle and synaptic terminal. Sorry, synaptic terminal of rod is known as spherule, right? And then yeah, and the outer segment it has discs. These discs contain rhodopsin, which is a visual pigment, and this visual pigment uh, basically it has rhodopsin. It basically gets bleached, and the entire procedure of image forming happens. That we'll discuss later. But for now, you'll understand that why is the outer segment photoreceptive, because it contains discs, and those discs contain the visual pigment called rhodopsin, right? And then you have the inner segment, you have nucleus, and then you have the synaptic terminal. Synaptic terminal of rod is known as spherule. Cone. Now, cone obviously in the front part it has a cone-like projection or cone-like shape, right? So this is the outer segment, and just like here it has this. Initially, these were membrane enfoldings, but then they separated and they formed disc. Here you have membrane enfoldings, and then you have the inner segment, then you have the nucleus, and the synaptic terminal is known as pedicle. Synaptic terminal of cone is known as pedicle. Right. So these are the rod and cones. And cones are fewer in number compared to rods. Rods are way more in number, like they're abundant, right? And another thing is that one cone it connects to only one bipolar cell, whereas multiple rods can connect to one bipolar cell, right? Okay, then we spoke about neuronal cells. So you have bipolar cells, and then you have ganglions. Then in the bipolar cell, this region is the dendrite. This one is the dendrite. Sorry, this is the dendrite, and this is the axon, right? And similarly in ganglion, this is the dendrite, and these are the axons. So basically, when they talk about optic nerve, or optic yeah, nerve basically, so it is made of axons derived from ganglions. So these are basically axons of the ganglions. They all collectively make up the optic nerve, right? Yeah. And then how did this thing come here? Right. So I was saying that, yeah, we've covered this also, we've covered this. Rods and cones and bipolar cells. Yeah, so rods and cones and bipolar cells, they generate graded potential. Whereas ganglion cells, these have voltage graded sodium channels, therefore those channels open, sodium influx happens, and then action potential is generated. So ganglion is capable of producing action potential, whereas bipolar cells and rods and cones, they can only produce the graded potential. Okay, moving forward, we have discussed this as well. And yeah, just to give a clarity, we took this region from here and magnified it here. Hence, why you can see that these are going in this fashion, right? I hope you can understand the similarity now. They're going into the optic disc and forming the optic nerve. So here, sclera, choroid, then the pigment epithelium, and after pigment epithelium, you have your photoreceptor cells. Then these are the bipolar cells in blue, and then the ganglions, and then the axons of the ganglions, they all cluster up and they form the optic nerve, right? Now, yeah, glial cells. Now, glial cells are non-neuronal cells in the nervous system that provide support, maintenance, and protection. Now, a very surprising fun fact, like you must be thinking that, yeah, neurons are a type of cell, right? And they must be really abundant when you talk about nervous system. Okay, you might be true, but in central nervous system, we're talking about central nervous system, the most abundant cell type is the glial cells, not even the neuronal cells, the glial cells. And in CNS, you have astrocytes, microglia, oligodendrocytes, you also have ependymal cells, and in peripheral nervous system, you have Schwann cells and satellite cells. So this is just for a recap so that you can, get a quick summary of the glial cells so that you can further link them. So astrocytes are most abundant glial cells. So I told you that glial cells are most abundant in CNS, right? And what sort of glial cell exactly? Astrocyte in CNS. We're talking about CNS. So what does astrocyte do? They have star-like shape and they provide support, nutrients, and they also form blood-brain barrier, a very important feature. Other than that, you have microglia. Now, microglia, they act as phagocytes of your CNS. Then you have oligodendrocytes. They basically form the myelin sheath around the axons, right? And similar to oligodendrocytes in PNS, you have this is in CNS, right? So in PNS, it's alternate. This is in CNS, so its alternate version is Schwann cell, which is there in PNS, which is peripheral nervous system. And yeah, so here microglia forms macrophages in CNS, then oligodendrocytes and Schwann cells, PNS and CNS, produce myelin sheath around axons. Ependymal cells, they form CSF in the ventricles of brain and spinal cord. And then you have satellite cells, also one of the glial cells of the PNS, and it forms cluster around neurons, and it is there for additional nutritional support. This was just a quick recap of glial cells, even though it doesn't fit appropriate here, but I just wanted to recap because we're also going to be discussing some glial cells here. So for that case, and we're just discussing astrocytes and microglia, but now you know that astrocytes and microglia, they are in CNS, and we are associating retina with CNS. Remember, I already explained you the reason why retina is linked to CNS. It's embryological basis, right? We've already discussed that this basically derives from neural tube, and neural tube, it gives rise to CNS. So that's how. Now, yeah. So in the previous lecture, until the previous diagram, we covered everything. Now the additions are in this diagram that I've added. Now, yeah. Also, RBGs. By RBGs, I mean these receptor cells, the photoreceptor cells, then bipolar cells, and then the ganglion. They take information radially from outside to inwards. And also in this diagram, I think I mentioned that from here. Current is moving radially from outside towards inside, from this layer to the out, like innermost. So outside, from the photoreceptors to the inside ganglion, and then to the optic disc. This is how the current is moving, or the information is moving. So yeah, and then but there's one thing that counters this, that is interplexiform cells. But let me go in order and then you'll understand this better. Other than these all layers, you have some additional cells, and sorry, other than all these cells, which I already mentioned: epithelial, photoreceptor, neuronal, and then glial. You have other cells as well. Let's first talk about um, HA and I. This is the mnemonic, which stands for horizontal cells, amacrine cells, and interplexiform cells. These are the cells that I never discussed last time. So photoreceptors, they communicate with each other also via local conducting cells. Lateral communication instead of radial. So we have radial communication which goes this way, but there are also cells present which allow these cells to communicate with each other longitudinally. So there's like lateral communication instead of radial, which is also present. And this is also present there. So that, wait, first let me explain the HAI cells. Where are they present? So here you can see horizontal cells, which form synapse with one rod and the other cone, or one rod and the all the other rods, or all the other cones and rods all together. So basically, they are horizontally placed here, right? You can see. So they basically form, they crosstalk with multiple rods and cones. This is the function of horizontal cells. Now, why is this present here? So that, for example, if any information is going like this, right? But there are chances that it might get, dissipated, and it might start going here up and down. So horizontal cell is there to take care of that. It's going to even grab that information and keep this entire feature enhanced and straightforward, and also so that cross-talking can be enabled between the photoreceptors. That, oh, okay, you found this information, I found this information. So this also allows the cross-talking, and also to keep a unilateral motion of the transformation of information. Then comes the amacrine cell, which is present here. So you just need to remember the placings. And these allow the cross-talking between the bipolar cells and the glial cells. So it comes here, right? And then it comes in the inner plexiform layer, and horizontal cells come in the outer plexiform layer, and then comes the interplexiform layer, which communicate between the outer and inner plexiform layer. So this basically takes the information from inner plexiform layer to the outer plexiform layer. So this is a function of interplexiform. So these are the cells that allow conduction to take place even laterally, and for interplexiform, between the plexiforms, and amacrine also laterally. Okay, so these are the cells. Now, so we discussed RBGs, we discussed high cells. Let's discuss the MAM cells. Now, since glial cells, I told you, they are there in neurons so that they can provide protection, and also now you see there are so many nerve fibers here. There's so many like ganglion axons here. Now we do not want any sort of short circuit here, because there's so many neuronal tissues in retina. Even here, this is also a neuronal cell. This is also a neuronal cell. There's so many neuronal cells. We do not want any sort of short circuit. So, and also one more thing, we want that these all cells, they remain intact and they remain in place. And also we want that there should be insulation, because insulation will prevent the short circuit, and we need to keep these things in place. For that very reason, we have the glial cells, and the mnemonic is MAM. Now you have M stands for Muller cells. Now what does Muller cells do? They hold the inner segment of rods and cones together, and it keeps them fixated in place. Now, this was the outer segment, right? No, we're not talking about outer segment. We are talking about the inner segment. So see here, that's the extensions of the Muller cell. Here, Muller cell is sitting right here, and it's sending its extensions, and they are keeping the inner segment of the rods and cones all together in place. It's ensuring that they all sit upright and in place. They're fixated. They do not move here and there, and there's enough insulation. There's no short circuit. And then this also forms a region. Here you can see these in yellow. It forms an interphotoreceptor extracellular matrix region, so that it doesn't mix with the rest of the region. So this region, anyways, we don't need to discuss for now because it's of a higher level. But yeah, It's there. And A is another glial cell which stands for astrocytes. Astrocytes, I've already told you, they are star-shaped cells. They formulate the blood-brain barrier, support nutrition. These are found there between ganglions and the axons, nerve fibers. And then you have the M microglial cells. Microglia, I told you, they act as macrophages of your CNS. Microglia, they can be found everywhere and anywhere. It's not specific to any region. Like glial cells, I told you that astrocytes, they're only found in between the ganglions and between the nerve fibers, right? And then I told you that Muller cells are only found between the inner segments of the um, rods and cones. Similarly, but not similarly, microglia, they're found anywhere and everywhere. They're just scattered. So now we've covered all the cell types: RBGs, then HAI, and then MAM. So you have the receptor cells, rods and cones, you have your bipolar cells, you have ganglions. Then you have horizontal cells, endocrine cells, interplexiform cells. Then you have Muller cells, rods and cones between the inner segment of rods. Then you have your endocrine cells, remember they're here. Then you have your, sorry, not endocrine cells, sorry, astrocytes. Remember they're here, astrocytes. And then you have your microglial cells, right? Okay, moving on. Don't forget that RB, they produce Berry potential, and G, ganglion, they produce action potential, and also neurotransmitters. If you talk about rods and cones and bipolar cells, they all are glutaminergic because they secrete glutamate as their neurotransmitters. So these are RBG. Whereas if you talk about horizontal and endocrine cells, they are inhibitory. They release GABA as their neurotransmitter, so they are GABAergic. And endocrine cells also secrete glycine and acetylcholine and dopamine. Yeah, so these are the type of cells that we have covered. And these are the 10 layers of retina that you need to have on your fingertips. Let's discuss them. First of all, retinal pigment epithelium. Then you have rods and cones, the photosensitive part, and rods and cones. But in this layer we are only discussing the photosensitive part, the outer segment and the inner segment. Sorry, just the photosensitive part, outer segment and the inner segment. But how is it that you can't include inner segment in this? Anyways, I think you're just talking about the photosensitive part, this region. So first of all, retinal pigment epithelial. Then you have the uh, a second layer, which is the rods and cones, the photosensitive part only, which is the outer segment. Then you have the outer limiting lamina, or outer limiting membrane, which is formed by the Muller cells here, outer limiting membrane. Then you have the neuronal, the outer nuclear layer, sorry, outer nuclear, gosh, outer nuclear layer, right? Which includes the nuclei of these rods and cones. So this is the outer nuclear layer. Then you have the outer plexiform layer, right? So what's the sequence? We've covered five layers by now, and these five layers, they're supplied by the diffusion of the choriocapillaries. Retinal pigment epithelium. Then you have the outer segment of the rods and cones only. So this is the outer segment only. Then you have outer limiting membrane. Then you have outer neuronal membrane, and then you have outer plexiform layer. Then comes the um, after outer plexiform, you have inner nuclear layer. Then you have inner plexiform layer. Then you have the ganglion. Then you have the nerve fibers, and then you have the inner limiting lamina. Inner limiting lamina is formed by the foot plates of the Muller cells. Muller cells has its extensions protruding in both the directions. Here it forms the outer limiting lamina, and here it forms the inner limiting lamina or membrane, whatever you want to call it. So these are the 10 layers. I hope they're clear. So that's pretty much it.